Hello class, this is Dr. A, and we need to finish up our discussions of secretion and reabsorption in the nephron, and this particular presentation is going to sort of recap the functions performed by the distal nephron, and I just basically mean everything past the macula densa. This is where hormones work, etc., etc. And then I'm going to talk in some detail about the renin-angiotensin-aldosterone system. So, as I just said, the distal nephron starts at the macula densa. You can divide the distal tubule into a number of small portions depending on what kind of cells they have and what kind of cells are acting on them. And then some of those cells spill over into the cortical collecting duct, and so sometimes to me it just doesn't make sense to memorize a lot of little portions when really the distal nephron is doing all kinds of things to maintain homeostasis depending on what kind of cell is available. Hormones can work in the proximal tubule. And actually a hormone that I didn't list here, angiotensin II, actually can work in the nephron to absorb sodium. We usually think of angiotensin II as a vasoconstrictor, but I'll get to that in a minute. So, in the proximal tubule, calcium can be reabsorbed in between the cells by paracellular transport. That's not going to happen in the distal tubule because the tight junctions are really tight. So if we need to reabsorb more calcium, what do? We use something called parathyroid hormone. So when your calcium levels are low, parathyroid hormone is released, and parathyroid hormone will help you absorb calcium, and at the same time you'll secrete phosphate. So if you hang on to more calcium, you have to get rid of phosphate, because calcium and phosphate can come together and form a solubility product, and you can get calcification of tissues, and that's not a helpful thing to have happen. Aldosterone works in the distal nephron, so the late distal tubule and the cortical collecting duct, there are cells that respond to aldosterone, and that promotes sodium reabsorption and potassium secretion, and I will talk about that in more detail in just a minute. Antidiuretic hormone, we've actually had a fair amount of detail on that. So in the collecting duct, there are ADH receptors, and those receptors activate a intracellular second messenger system that causes the insertions of aquaporins and increases the water permeability of the collecting duct so that more water can be saved, and as a result less urine is produced, and the urine that is produced is highly concentrated. We've talked about ateronatriuretic peptide in terms of its effect on the glomerular filtration rate. It's going to dilate the afferent arteriole and increase the GFR. It's also going to work throughout the tubule and in the distal tubule to inhibit or decrease sodium reabsorption. So if you have more filtration and less sodium reabsorption, then more urine and more sodium loss is going to occur. This would be appropriate if you had too much plasma volume. Angiotensin II comes from the renin-angiotensin system. Angiotensin is a vasoconstrictor, but it also works throughout the nephron to increase sodium reabsorption and increase blood pressure. It's important to think about the gradients across the distal nephron. So you've been through the proximal tubule, and the proximal tubule is reabsorbing about 66% of the sodium. And then maybe the loop of Henle does another 20%. So 86% of the sodium has been reabsorbed. And the early distal tubule has some cells that reabsorb sodium and chloride, and this particular form of transport, water does not follow, so it's not isoosmotic, and this is worked upon by thiazide diuretics. And then later on in the tubule, we can have the effect of aldosterone. But if you've reabsorbed all this sodium, and extracellular fluid sodium is 140, something like that. Could be up to 145, but you know, let's 135 to 145, let's say. And you've reabsorbed a whole lot of that sodium. If you have leaky tight junctions, that sodium will leak back into the filtrate, and we just can't have that. The other thing I want to remind you about is that the proximal tubule does some hydrogen secretion and bicarbonate reabsorption. And remember, in the proximal tubule, hydrogen ion is secreted by secondary active transport. Well, when we get to the distal tubule, again, we're not going to have a lot of sodium left, and the hydrogen gradient is fantastic. It is huge. So then further hydrogen secretion has to occur by primary active transport. Again, the reason why we need these very tight junctions is that we don't want hydrogen leaking back into the body. So my nickname for the distal nephron is the fine tuner, because the distal tubule is not about quantity, it's about homeostasis. So fine-tuning reabsorption and secretion depending on what your body needs. So the proximal tubule and the loop of Henle reabsorb potassium if you need to get it from your diet. But if you have too much potassium, the distal nephron is going to secrete potassium, and that's going to be with the help of aldosterone. It's going to secrete extra hydrogen ion, and that's going to be the help of that ATPase. It's going to reabsorb extra calcium. That's going to be with the help of parathyroid hormone. And it's going to reabsorb extra sodium. In the early distal tubule, it's the sodium and chloride transport. In the late distal tubule and early collecting duct, it's going to be an aldosterone mediated mechanism. Also remember that beginning at the distal tubule is the macula densa, and the macula densa can tell the afferent arteriole about sodium chloride load at the macula densa. If the load is low, it means the GFR is too low, and we want to dilate the afferent arteriole to get the GFR back up. If there's a whole bunch of sodium and chloride showing up at the distal tubule, that means the GFR is too high, and the afferent arteriole is going to be constricted. So I'll make a dilated versus a constricted to lower the GFR and get the level of work at the distal tubule back into check. So a really big important hormone system that we talk about in the kidneys is the renin, aldosterone, angiotensin system, and that's what we'll spend the rest of our time on. So, at the juxtaglomerular apparatus, remember, you have an afferent arteriole, and you have a macula densa, and they can talk. And this whole thing is called the juxtaglomerular apparatus. And there are some cells around the afferent arteriole called juxtaglomerular cells, and they produce renin. So, the macula densa talking to the afferent arteriole can be the basis of tubuloglomerular feedback, but it also can be the basis of blood pressure regulation. So if your blood pressure is low, you're going to tell your juxtaglomerular apparatus about it in 3 ways. First of all, the sympathetic neurons. So if the baroreceptors are less stretched, then you tell your brain that you have less blood pressure, and they activate the sympathetic afferents. And sympathetic afferents go to those juxtamedullary cells, and we get renin. If the afferent arteriole is less stretched, remember, according to autoregulation, we'll get dilation of the arteriole. But if your afferent arteriole is less stretched, you're going to produce renin. So that's the second thing here. And if your blood pressure is low, then your GFR is going to go down, and if your GFR goes down, there'll be less sodium chloride at the macula densa. That is going to tell the afferent arteriole to dilate, but it's also going to tell the juxtaglomerular cells to produce renin. Renin is an enzyme. It's not a hormone, it's an enzyme. And when it goes into the blood, it finds a gigantic protein, actually it's not listed here, a gigantic protein called angiotensinogen. Now this ogen means angiotensinogen is inactive. Renin gets us to angiotensin 1. Then we have another enzyme called ACE that gets us to angiotensin 2. Angiotensin 2 is the active ingredient here. And angiotensin 2 is going to do a number of things. It's going to tell you you're thirsty. It's going to constrict your blood vessels. It's going to tell you to reabsorb sodium. And it's going to go to the adrenal cortex and tell the adrenal cortex to release aldosterone. Aldosterone is going to go in the blood, it's going to go in the late distal tubule and the cortical collecting ducts and tell it to reabsorb sodium. Later in the tubule, water will follow. It's important to realize that sodium and water are not traveling together. Sodium travels first, and then other cells that have aquaporins will allow the water to follow. So if your blood pressure is low, you make angiotensinogen, which is a vasoconstrictor that will raise your blood pressure, and you produce aldosterone, which causes the reabsorption of sodium and the reabsorption of water later in the tubule. That increases your plasma volume. So you're going to increase your vascular resistance and increase your preload. That's going to regulate your blood pressure. So aldosterone is a steroid. And we are going to talk about steroids later in the semester, but maybe you learned in some of your other courses that steroids promote gene transcription. So aldosterone is going to find an aldosterone receptor. Aldosterone and its receptor are going to go into the nucleus, cause the transcription of genes that are going to support the kind of transport that we want. And what are we going to get? We're going to get a sodium-potassium ATPase, and maybe I'll get Lucille back here. So we'll get a sodium-potassium ATPase, so we'll promote a bigger gradient, and then we will have two kinds of channels. So we'll have some ENaC channels, or epithelial sodium channels, and because the gradient for sodium is in, we're going to promote sodium reabsorption. And we're also going to have some epithelial potassium channels. And these potassium channels are then going to allow potassium to leave via that same concentration gradient. So aldosterone is going to promote the action of these particular proteins. I want to say these cells are called principal cells. I always have a hard time remember the names of cells, but I think these are principal cells. And if we promote the ATPase and get a bigger gradient, then we promote potassium secretion and sodium reabsorption. Now, aldosterone not only regulates plasma volume and blood pressure, but can directly regulate plasma potassium. So when your plasma volume is low, and as we showed, as we showed in this diagram here, if your blood pressure, plasma volume is low, sympathetics, less stretch of the afferent, a decreased GFR and less flow to the macula densa, all of those things are going to tell your kidney to produce renin, and you get angiotensin 2, which is a vasoconstrictor, and aldosterone will promote sodium reabsorption and potassium secretion. Later on the tubule, water will follow sodium, and that increases your plasma volume. But please note that potassium secretion is also going to occur. So notice that an increase in plasma potassium will directly activate the adrenal cortex to release aldosterone to promote potassium secretion. And why is it important to regulate potassium? Well, I hope you all remember your Nernst equation and resting membrane potentials. So, if potassium gets to be too high, that has a depolarizing effect on the resting membrane potential, and that can be deadly for the heart. So this is an important negative feedback mechanism. If, of course, there's been a large insertion of potassium into the situation, let's say you had muscle trauma and you have a whole bunch of potassium being released, this mechanism might not be able to keep up with that, but it does have some role. Now, I was thinking I had another diagram, but I don't. So, remember, we are finishing up sort of normal physiology, so we'll get all the way to the distal tubule. And then the next week, we will work through the problems as a class, so all those clearance problems and renal fraction problems, and then we'll talk about renal pathophysiology. And then the week after that, we have our renal exam. So thank you all for hanging in there with me. Thank you all for listening, and I'll see you in class next time.